The overall goal of this procedure is to develop, apply, and demonstrate a detailed procedure for library-based bacterial identification using MALDI-TOF mass spectrometry. This is accomplished by first acquiring mass spectra of bacteria using a MALDI-TOF mass spectrometer. In this study, mass spectra of 15 bacteria isolated from a karstic environment are collected. The resulting peaks form characteristic patterns or fingerprints of the bacteria analyzed. These mass spectra are then imported into software and a model database is constructed. Next, the reference mass spectra in the model database are processed and analyzed using cluster analysis, peak matching, and statistical analysis. Finally, mass spectra of blind-coded bacteria, which are randomly selected from these 15 bacteria, are collected again and compared to the reference spectra in the model database for identification. Results show that bacteria can be correctly identified either based on similarity coefficients or potential biomarkers and peak classes. Begin this procedure with deposition of 1 microliter of protein extract containing no viable cells onto a stainless steel MALDI target.